This meeting.
Okay, so we were in the middle of chapter 1, and so this is a lot smaller than it usually is. So squint, I guess. I don't know. Not quite sure why the screen. I guess some sort of a zoom setting on there. That's a little bit different. Okay. So basically, we're looking at the body parts, right? And so we're going to be taking a look at regions now. And so first of all, a couple of regions that you're going to see is the names of the body, the regions of the body are essentially listed, and the body parts are oftentimes mimicking the regions, right? And so that's the reason why we do regions. So for instance, the head region typically is cephalic in general, neck is cervical, you have oral, nasal, orbital, right? So that is mouth, nose, and eyes. And then, of course, in the thorax, or the trunk, if you will, you have basically kind of underlying structures associated with that particular area. Like, for instance, in the middle of your chest where your sternum is the sternal region, the pectoral region, which is where your pectoral muscles are, and things of that nature. Abdominal, pelvic, and things of that nature. Now, on the right-hand side, this is the one, those are the easy ones, right? So those are the ones we probably already know. The ones that are a little bit more tricky are the ones that are on the right-hand side. So these ones that, generally speaking, oftentimes we know some of them, but maybe not all of them. For instance, an easy one would be the clavicular region, where your clavicle is, it's your collarbone, right? So axillary is armpit, brachial is arm, antebrachial is the elbow region. So remember, these are regions, not like the actual elbow itself. These are just the regions, the elbow region itself. And so to me, antecubital is elbow, antebrachial is the forearm. And then you have carpal and so forth. When you get into the legs, it gets a little tricky because you have different different names for the different pieces. First of all, the underlying bone, for instance, femoral, the knee, which is patellar, the shin bone, or the tibia, is the crural. The sural is the backside of the leg itself. And so oftentimes you learn these regions because this is what structure is going to be named after. It's going to be named after these regions. So your palmar arch, for instance, is a vascular kind of an arch structure that's in the palm region, or the palmar region of your body. That's in the hand. And so those are some of the areas. If you turn yourself back, you'll see you have the occipital region. Why? Because that's where your occipital bone is. The back of your head is the occipital region. The nuchal region, which is the back of the neck, because typically the nuchal is associated with the lines of the skull on the occipital bone itself. So there's like nuchal lines or nuchal ridges that are right there. So that kind of defines that region. The lumbar region, the vertebral region, so the thoracic region, or the cervical region. So these are all regions of the neck, trunk, and lower back. As you start to get to the back of the leg, you get to the popliteal, which is the back of the knee. Patellar is the front of the knee. Popliteal is the back of the knee. Crural is the front of the leg. That's the tibia. The back, where the gastrocnemius is going to be the sural, right, or the calf region. And then, of course, you have things like calcaneal and plantar. Some of you guys may have heard some of these, right? So if you have like plantar fasciitis, that is associated with the sole of your foot. It's the connective tissue ligament basically associated with some of the muscles that wrap underneath your foot and connect to the bottom of your foot. And those tend to be inflamed. And that's what plantar fasciitis is, typically very difficult to deal with, especially if you're an athlete, to be able to walk and put pressure on that because it's not feeling very good. And so if you're into athletics, you follow athletics, you know that the plantar fasciitis injury is one that takes a long time to get over. And it's an easy one to re-aggravate. And there's actually many stories of many athletes who in their hurry to get back on the field, rush things a little too soon, and they just re-injure the same thing. And sometimes it's been a career killer. There's several Rockies players that have gone to that particular fate. Okay, so let's take a look at the regions of the body. So this is inside the body. We already talked about the regions of the body, but now we want to take a look at the inside. So we're going to basically take a look now at your cavities. So we have a couple of different cavities, I suppose, ventral and dorsal cavities. The main ones are going to be the thoracic cavity, abdominal cavity, pelvic cavity, which are the ones we tend to focus on in AMP. But what we have is we have essentially two systems to divide up the abdominopelvic cavity. One is kind of a quadrant system where you have upper and lower quadrants, so right, left, upper, and right, left, lower quadrants, which you can see here. Typically, it's kind of bullseyed right in the middle of the small intestines, so just a little north of the appendix of the large intestines, kind of shifted over, kind of right in the middle of that area, and so that kind of defines your quadrant system. And the other one, which is a little bit more kind of teased out, is going to be sort of what I call the 9 grid. So it's basically 9 different, I guess it's not quadrants at that point, it's what, non-grants at that point, I guess, I don't know what the term for 9 is. Right, so basically there's 9 of these now, so still kind of targeted right in that same region, which is right dead center of the small intestines. The bullseye, the middle of the tic-tac-toe grid, is what's called the umbilical region, so named after the belly button, right? Because that's basically, actually belly button's a little further south than that, but that's what it's referred to. And then above and below that, you have directional terms. For instance, you have epigastric just above that. Epi means upon, gastric, the stomach. So this is upon the stomach, so you can see the stomach underlies this particular region. And then hypogastric, which means below the stomach. You can see this is going to be below that particular region. And then you have the lumbar region, right and left. The iliac region, both right and left. And the iliac is associated with your ilium. So that's your hip. So basically your iliac region, you're talking about your hip girdle right here, right around in this area, kind of at just a little above your belt line is essentially what we're talking about at that point. Okay. And so you have different structures there. And of course you have right and left hypochondriac. That's not somebody who steals, right? This is, so hypo means below, chondro means cartilage. So this is below the rib cartilage associated with your ribs. So this is the right, basically just below the ribs region. That's what the name means. And this is the left, just below the ribs region. That's what this name means. Okay. Now the reason why you do this is because this gives you a sense of sort of geographical location in a patient. Imagine if you're a surgeon and you need to make a cut to find an organ. Well, you're like, okay, so, you know, where is everything in here? So if you have a grid that you can draw on a basic patient, then you can kind of essentially locate where roughly these different organs are, so that if you're going for the appendix, for instance, you don't need to be making a cut up here in the left hypochondriac region, because you know the appendix is going to be further down here at the boundary roughly of the right iliac versus the right lumbar, okay, in that range. And so that's the reason why you have this, is so that you can locate roughly where these organs are before you just go hacking into somebody, right? Because surgery can get quite messy if you're just sort of butchering the patient. And so we learned that one the hard way. You know, you enter someplace and you're like, oh wait, it's not here. Where is it? Okay, let's try over here. You know, it's like, nope, it's not there either. Let's try over here. So before too long, you basically killed your patient, right? And that's kind of where we started way back in the day, probably working on gladiators, because after all, I mean, gladiators, we pay them to die, essentially, is what they did back in those days, right? So they're going to die anyway, so why not just cut them up and get something useful out of them? That's what Galen, who's kind of noted as the sort of preeminent doctor, founding doctor of medicine, if you will. He's the one who worked on gladiators and kind of got kick-started this whole anatomy and physiology area, way back in the day, working on gladiators. So the other thing we want to do in terms of getting our aspect down is taking a look at the orientation of the body in terms of the planes of the body. Now, these are important, especially when you're looking at imaging and things of that nature, right? Because you have to get the image in a particular aspect. And so we have a couple of different types. So first of all, a sagittal cut or sagittal plane is one that's going to basically cut your body into left and right portions, right? So it's going to be left and right. So this is going right down the middle of your nose, right? That's your median line. So that will be a sagittal cut. And that's true for anywhere you cut. It doesn't have to be down the midline. It can be anywhere. So that would be a sagittal cut. Typically at the mid of the sagittal plane is your median or the midline of the body. So generally speaking, we usually think of cutting the body down that midline or down the median. Now the frontal is a little bit different. So you're going to turn this 90 degrees. And in this case, you're going to cut the body into anterior posterior. This is oftentimes called the coronal. So you can think of the corona, right? So kind of like the corona of the sun, kind of like a little halo. So if you cut like this from ear to ear over the top, that would be a coronal cut. So you're creating a front and a back aspect, the anterior posterior aspect. And then a transverse is a little bit different. In this case, you're going to be cutting into superior and inferior section. This is basically a cross section. That's what this is. So basically, if you take me at my waist and just cut me in half, then that's a cross section. And that would be a transverse cut. There's another one, oblique. This one is an angular cut. Typically, it's a non-right angle cut. So if you were to take kind of a blade and sort of cut me diagonally across my body, that would be an oblique cut. Okay? And typically what would happen is that the cross section would oftentimes be kind of looking something like that. In like a round, that's kind of like the bone, cross section of the bone. If it's oblique, it's going to look more oval on the end. And so these are important because depending on how we cut the body and in imaging, like in a CT scan, for instance, when you're doing those slices of the body, you got to know whether or not you are doing either sagittal slices or frontal slices, right? Because that's going to determine your orientation about what you're looking at. And so as you kind of get into some of these, it will make a lot of difference. And you can see slightly different things, right? So here's a good example. I love these images, right? So here's a transverse section. You can see you're just kind of slicing her head off right there. And so this basically gives you this transverse aspect of the brain, as if you're kind of looking down through the top. And so we've seen a lot of these, right? So we oftentimes see a lot of these in different images. So this is a very, very common one, especially if you're doing like an MRI, because what will happen is you're laying down in your supine position, and the images are basically taking slices of your head like this in a transverse aspect. And so that's kind of what you see. You kind of zoom in through the head from the top, and then you kind of go down through the rest of the brain if you're doing like a brain MRI. Okay. Now, if you do a coronal cut or a frontal cut, this is basically what you're doing. So essentially you're taking it straight down and kind of cutting through your face. These are really cool, not as common, but really cool. Now, for the brain purposes, we'll see these oftentimes as well. Notice you see different aspects. You can see the lateral ventricles, you can see the third ventricle, and you can see the white and the gray matter of the brain itself. And so a lot of times you'll see this, especially these two aspects when you look at like neurological imaging. You'll oftentimes see those when you're taking pictures of, say, for instance, Alzheimer's brains and things like that, to see if you can see abnormal atrophy or structural decay in the actual structure of the brain itself. And sometimes you can pick that up with these particular images. Then for the sagittal section, you're going basically straight down the middle. And so this is kind of like what our models are. So most of our models are kind of in this sagittal aspect when we take a look at the brains themselves. And it's really good because you can see kind of a side-on view of a lot of the structures that are associated in the brain itself, especially that middle portion of the brain, where you start to get to the thalamus and things of that nature. Okay. So you can see that each of these different sections gives you a slightly different view of the overall organ. Remember, the organ itself is three-dimensional. And so having these different views allows you to be able to see things from different perspectives and it'll gain more information than you would otherwise get if you were just using one particular view. Okay, now that's kind of planes to the body. Now, what about planes through an organ itself? Well, there are a couple that we see a lot. So we have what's called the longitudinal plane. This is basically when you cut an organ sort of lengthwise, if you will. So you kind of cut it along its long axis. And so that would be a longitudinal cut. A transverse or a cross section is basically when you cut it perpendicular to the length of the organ. They're kind of using intestines here as a good example. And then you still have oblique with this one as well, where you're basically cutting it at an angle. So here you can see you have this kind of long cylindrical looking structure in a longitudinal section. And then other times you see a transverse or a cross section, which gives you the kind of that perfect view just sort of through the end of this particular structure. And then of course the oblique will come across more of like a little oval. You don't see oblique sections very often because, to be quite honest with you, there's nothing that oblique can show you that you can't already get with a cross section. Okay. And so they're a little rare, and they're a little harder to interpret. Sometimes they will kind of throw students off a little bit when they're taking a look at an oblique because it'll look weird, and you're like, what am I looking at? So it's easier. Most of what you see, like in histology, for instance, are just longitudinal sections and cross sections. Okay, so let's take a look at the body cavities. So the body cavities that we basically have, there's a couple of major ones. So we have both dorsal cavities and we have both ventral cavities. Remember, dorsal is your back, posterior, ventral is anterior, that's your front. And so the cavities that we have basically or dorsally are essentially these guys here. So these are the dorsal ones. So you have your cranial cavity, that's basically your skull and your brain inside your skull, and of course your vertebral canal where your spinal cord will go through that vertebral foramen, which you guys will be seeing tonight during lab. And so that would be considered a cavity once your vertebral column is in place. Okay, and so those would be considered your dorsal or your posterior cavities. Now we deal with that a little bit, but really the ones we really focus on a lot are the ventral cavities, the ventral body cavities, the thoracic cavity in particular, and the abdominopelvic cavity, which is kind of a combo. We oftentimes will toss them together. So when you take a look at your thoracic cavity can be broken up into two subsets. Well, three actually, if you think about it. So first of all, you've got the pleural cavities. So here's your thoracic cavity. Looks like a giant bell. So your pleural cavities, which will be on either side, houses your lungs. So these will be the pleurals. In the middle is what's called your mediastinum. So that's this little middle central portion. And the lower portion of that is going to be your pericardial cavity, where your heart is. Up here, this is where your esophagus and your trachea are, in the upper mediastinum. And then below your thoracic cavity is your abdominal cavity. So this houses your intestines, your stomach, and so forth. And then at the very bottom of your abdominal cavity here is going to be your pelvic cavity. So your abdominal cavity contains all your digestive organs, Your pelvic cavity is basically where your reproductive organs are. For instance, the lower portions of your digestive, like the rectum, your urethra, your bladder, and also the reproductive organs. For instance, if you're female, this is where you will find your uterus and your vagina. For males, this is basically where the prostate will be, and all those glands associated with creating semen. Those will be in that sort of lower region. Now, there's not a boundary. There is a boundary for thoracic and abdominal. It's called the diaphragm, but there's not a boundary for abdominal and pelvic. It's just kind of a big chamber, and then it's the lower portion is the pelvic. Okay. So here's that. This is kind of what it looks like. So here's your dorsal. You can see it's on your back, and then of course your ventral or your anterior will be broken into your thoracic, your abdominal, and your pelvic. And then this is really good because you can see your mediastinum and your pleural. So here's your pleural cavities. This is your pericardial cavity. And this is your upper mediastinum. And then you have your big diaphragm right here. And then you have your abdominal cavity right here with all of your organs in it. And then the lower portion will be your pelvic cavity. So when you take a look at the cavities, we have to take a look now at how we organize them. And so I'm going to switch back to the smaller version because so when you're taking a look at cavities, you have to line them, right? You have to line them and you have to protect them. That's going to be a subject of chapter 4. We're going to be taking a look at the tissues, and we're going to be taking a look at the membranes. But ultimately, you have several different types of membranes. You have cutaneous membrane, mucous membranes, you have serous membranes, synovial membranes, and so forth. The serous membranes are the ones that are important, especially for the organ systems, because these are the liners of not just the organs themselves, but it's also the liners associated with the cavities. And so when you take a look at serous membranes, there's basically a similar sort of a repetitive system that you see throughout the body. The first one, which is called the visceral serous membrane, is the one that covers the organ. So this is the membrane that's resting right on top of the organ itself. Okay. So then from there, there's going to be a little bit of a gap or a space, generally speaking. And then you're going to have what's called the parietal serous membrane, which essentially lines the cavity. And so generally speaking, what will happen is if you were to take a look at the cavity, the wall of the cavity has this parietal membrane on it. And that's kind of like, it's like a liner of wallpaper, right? And then you'll have a space, typically called the serous cavity, or so like the pleural cavity is what it is in the lungs. And then you, on top of the actual organ itself, will be another like very thin layer, a very thin membrane. That's going to be that visceral layer. And so when you take a look at an organ, in particular the heart itself, and we do this oftentimes for the heart to understand it and its layers, is it's like putting your fist in a balloon, right? So if you put your fist in a balloon, what's going to happen is it's going to form two layers in a cavity, right? The part of the balloon that's making contact with your fist is going to rest right on top and wrap around your fist. That would be like the visceral layer. Then there's going to be a little airspace, and then there's going to be the outside of the balloon, right? The rest of the balloon, and that would be like the parietal layer. And so that's basically what you get. So for instance, this piece right here, this basically would be the visceral layer because it's resting on top of the organ. Then you have your cavity in between. And then on the outside would be your parietal layer. And that's a common pattern that we see in all organ systems. They have that same kind of serous membrane patterning. And we call it by different names in different organs. For instance, in the heart, it's basically the pericardial, the pericardium. These are the serous linings that surround the heart, right? One is resting on top of the heart. There's a pericardial cavity filled with fluid. And then there's the parietal pericardium, which is on the outside. It forms kind of like a little cellophane baggie, the pericardial sac, around the heart itself. And then that's what's located inside the pericardial cavity. And you can see it right here. So they basically placed it inside that pericardial cavity. The pleura, or the lungs, has the same thing. It has the visceral pleura, which is the membrane that rests right on top of the lungs itself. And then it's going to have a pleural cavity, which is this open piece right here. And then it's going to have this dark blue line, which is going to be the parietal pleural cavity, or the parietal pleural layer. And in the digestive system, it's called the peritoneum. Now, the peritoneum is basically the same thing. It's a serous liner. So there's going to be a lining on your digestive organs. There's going to be a gap or a space, and then there's going to be a liner against the wall. Okay, so you have parietal peritoneum and you have visceral peritoneum. And so with the peritoneum, it's a little bit more complicated because you've got more organs in there to deal with. And so when you take a look at it, what you have, and I want to present this one because I want to blow this up now, a little bit bigger than it actually is. So what happens is you still have organs and you still have this visceral layer that's wrapped around it. So that's going to be this kind of reddish purple layer that you see right here wrapped around it. The blue layer is basically going to be the parietal, or the outer layer that's on the cavity itself. Now, the important thing about the serous membranes in the digestive system is it basically wraps not just one organ, but it wraps all of the organs. And in addition to that, it kind of creates these flaps and folds. For instance, the visceral layer will actually extend from the liver and connect to the stomach in what's called the lesser omentum. And then what's going to happen after that is you will have another branch of this layer, this visceral serous, that's called the mesenteries, creating what's called the greater omentum, and it's a flap that will connect to all of the intestines. So these are cross sections of your small intestines. They're all held together by these little flaps of serous membrane, visceral serous membrane. And so your serous membranes are actually holding all of your digestive organs together. Okay, and this kind of sheet-like web that's all bound in, called the mesenteries. So the mesenteries basically are this kind of like webbing. So you can see here's the small intestines, and you can see this green part is the mesenteries. That's that sheet-like membrane that's connecting all of the small intestines together. Okay. So you don't just have your intestines sort of free floating around in your cavity. It's actually all bound up and sort of bundled up together with these sheets of membrane, serous membrane, called mesenteries. And so those are important. So this is the kind of the blab slide mesenteries. We just talked about that one, right? So the reason why these are important is because they don't just bundle together your digestive organs, but they also act as a pathway for blood vessels and nerves. So these will actually, so your nerves don't have to figure out how to get there. They just travel through the mesenteries. So it's kind of like a way to get everything connected and sort of networked throughout your digestive system so you can communicate with your digestive system efficiently. There's also what's called retroperitoneal. So retro means behind or back, behind the peritoneum. And so retroperitoneal means basically that you are behind your kidney, your peritoneal layer. So for instance, here you can see the parietal layer, which is the blue part, but you can see there's organs that are behind that layer, like your kidneys, for instance, reside behind the parietal serous layer. So does your adrenal glands. Those are referred to as retroperitoneal, behind the peritoneum. Okay. So what happens if you inflame these? Then you get nasty things that we don't want to get. Things like pericarditis, right, which is the pericardial inflammation, pleurisy, which is pleural inflammation, and then peritonitis, which is peritoneal inflammation. inflame your serous membrane because typically bad things happen when you do. Okay, so that's chapter 1. Okay, no, I don't want that. But as is the case, there's no rest for the weary. We're on to chapter 4. If you're wondering, chapter 2 and 3 is just basically a general bio review. And so since 1080 is a prereq for this class, and that means you already have a little bit of general bio under your belt. Okay. So let's go ahead and take a look at chapter 4. Chapter 4, we're going to be talking all about tissues and histology. Yes. The PowerPoint, you mean?
Yeah.
Okay. It could be that maybe what happened was I forgot to turn on that module. That is a possibility. Let me do this here real quick. That's exactly what happened. Okay, now take a look. And I don't know why Canvas does that, but whenever they create a module, they like automatically shut it, like make it invisible. And none of that, but they decouple the stuff that you put in there that's visible is different than the module. So you can have stuff in your module that's visible, but your module will be closed. So it's like you have to open up both. I don't know why they do that exactly. It just drives me nuts because there's like 2 different things to keep track of. And that's a pretty common thing. So if you don't see it, just let me know. And it's probably just the module. I didn't turn that on. Okay, let's take a look at some tissue and histology. So this kind of goes along with our lab that we did last week. And so ultimately, when you take a look at tissues, the first thing we want to do is you want to essentially define tissues. And so in 1080, we learned a little bit about cells, right? We learned a little bit about something in cells called extracellular matrix. And so we learned that a cell will basically secrete molecules outside of itself, things called like ground substance and things of that nature, which are polysaccharide types of gel-like consistency matrix that's out there. Plus, they'll put a bunch of protein fibers out there like collagen and elastin and things like that. And they'll create basically an environment for themselves that is helpful for their function and kind of creates a sort of a sense of, you know, like a sense of home, if you will, but also functionality. Now, when you take a look at tissues, that's an important concept because in some cases, your tissues are defined by the cells, like epithelial tissue, for instance. And in other cases, the tissues are actually defined by the extracellular matrix, like connective tissue. Right? Most of what we think about as bone tissue is actually extracellular matrix of bone cells. And so in some cases, they're characterized by one or the other. Now, when you take a look at tissues, tissues basically fundamentally are just cells of a common type that are fastened together in sheets to do a common function. That's basically what they're designed to do. And we have 4 major types of these tissue types. So we have epithelial tissue, we have connective tissue, muscle, and nervous tissue. Now, generally speaking, histologically, whenever you take, say, for instance, like a biopsy, right, and you sort of take a look at these, this is kind of what you guys were doing last time. So when you guys are doing histology, taking a look at tissue samples and studying them and trying to figure out what the different types of tissues are, that's histology, okay, a microscopic study of the tissues. And generally speaking, you need a biopsy. So like all the samples you were looking at came from biopsies. Probably a lot of them were cadavers, but they were all biopsies from like real organs and real people, okay? And so oftentimes we'll send off a biopsy for analysis and to kind of tell us, you know, hey, let's send it to a pathologist whose job it is to take a look at tissue samples and determine whether or not they're normal or abnormal, and then what are they, and then the doctor can go from there. Ultimately, when you take a look at sort of our tissues, we also do that during autopsies, right? Because one of the purposes of an autopsy is not like a cadaver prosection. You're not just there to look at stuff. Typically, the ones doing an autopsy, a medical examiner and their staff, are already well functioning in terms of human anatomy. So they know what they're looking at. And their focus is a little bit different. So rather than looking for things, they're looking for a couple things. First of all, they're looking for cause of death. Right, so they're kind of like it's a little bit of an investigation. So why did this person die? And sometimes that's fairly easy, especially if you're like a heart patient who's suffering from congestive heart failure and, well, it just finally gets the better of you, to like accidental or unforeseen deaths where it's like, well, we're not quite sure why this person died. They shouldn't have died. They seem to be in good health. So you have to take a look at that and do an autopsy, figure out cause of death. Why? Because on every death certificate, you need cause of death on the death certificate itself, mostly because, not just because people want to know, I mean, that is one thing, but also because the police department needs to know whether or not this is something they need to investigate, right? So depending on the cause of death, they could be needing to open up a potential homicide case, okay? And so those are the sorts of things that you're doing when you're taking a look at tissues, especially toxicology situations, how tissues kind of respond to different types of toxins and things of that nature. So there's a lot of things you have to do when you're doing an autopsy. Now, generally speaking, all of your tissues in your body all begin to lay down during embryology. So once we all start from the same place, we all start as a single cell. And then we go through a series of cell divisions to become ultimately big old head in there. A little baby. All right. In this case, I drew the diaper, but diapers aren't included. Just to let you guys know, in case you're wondering, they don't come with diapers. So on the journey there, what you're doing is a couple of things, right? Embryology is big and it's detailed and it's complex. And I don't want to go through the details of it, but I'm just going to give you a quick sort of a summary of what's happening during this period, right? So this is your 9-month period between when you are a single cell to when you are born. The first things that are happening is, number one, is you need to make a big multicellular body, which essentially means you need more of everything, right? You need more cells because your body now has trillions of cells in it and you just need a ton of cells. One cell is not going to cut it. That's the reason why you're going through lots of cell division. You're just making tons and tons and tons and tons of cells. Now, the other thing you need to do is you need to organize those cells. And some of those early decisions that you make in embryology are critical ones, right? For instance, you have to, first of all, determine where's the head and where's the butt, right? Where's the front? Where's the back? Where's the right? Where's the left? So all those axis determinations are all happening very early in embryology. Generally speaking, you're a little cluster or a little ball of cells when those initial decisions are happening. Now, the other thing that also happens is you have to start building tissues because you can be a little ball of cells, that's all well and good. But at some point, you have to start determining that this little group of cells here are going to go off and become nervous tissue. And these guys are going to become muscle, and these guys are going to become skin and so forth. But before you do that, you break them into what's called the germ layers, right? So in embryology, we have what's called the germ layers, which basically are 3 different layers that are the foundation for all the other tissues in your body. So these basically form all the tissues in the body. So and there's 3 of them. So we have the endoderm, which is the inner layer. Now, the idea of derm versus you're talking about a ball of cells that start to sort of invaginate. You're starting to get this little kind of what's called a coelom. That's like a cavity in there, right? And then what you have is this sort of notation system in your words of inside versus outside and so forth. And that's what it's referring to, that little ball of cells. And I don't want to get into that too much because that really starts to have to kind of flesh out a lot of development in order to understand like what's the derm and what are they talking about. You have to really see the structure of the blastocyst in order to really understand what's going on there. But necessarily, this is the endoderm is an inner layer, and so it forms typically or will grow up to form parts of the digestive tract and its derivative pieces. Okay, liver or pancreas, things like that. The mesoderm, which is the middle, meso means middle, right? Typically, this will go on to add its functions to the muscle, bone, blood vessels, things like that. The ectoderm, which is the outer part, will form the skin, and the neuroectoderm, which basically becomes the nervous system, so the crest cells, which will kind of move around, and there's a lot going on with the formation of your spinal cord. Skin pigment cells, adrenal glands, face tissues, things like that. So these will kind of all add their piece. They'll all grow up to basically add pieces to that. Now, it's not as easy as saying, oh, well, the endoderm leads to this, and the mesoderm leads to this. Because actually, when you take a look at most structures in your body, most organs in your body, they don't derive from just one of these. Oftentimes, there's a combination of these. So these three will oftentimes add bits and pieces together to create an organ or a particular type of tissue. So most of your organs in your body are originating like an amalgam of these three. So these three typically have inputs on all of it. So you can't just say, it's like, oh, well, this one only does muscle. Well, there's muscle that also comes from the ectoderm as well. Okay, so they kind of add their piece together. Now, that's kind of the beginning, the foundation. That's where kind of it all comes from. So the first of the four, right, is basically going to be epithelial tissue. Now, the epithelial tissue is a really important tissue. This is what you guys spend a lot of time on. Connective tissue is also pretty big, but epithelial tissue is pretty big. This is going to be a tissue that's dominated and is described by its cells, right? So not its extracellular matrix. Because basically what epithelial tissue is are essentially cells connected into sheets. Think of like a page of stamps. Right? Each individual stamp is like an individual cell, and they're all connected together in this big sheet of multiple stamps, kind of all connected to each other. Generally speaking, epithelial tissue has two primary and dominant functions. The first one is it covers stuff. It's a liner. Generally speaking, it's going to cover these body surfaces, both inside and outside. The other one is it also forms glands, which basically means it's secretory as well. Those two. Oftentimes in A&P, we remember the body covering and the liner piece of it, but sometimes we forget about the glandular piece of it. Okay. So a good example is the skin, right? Our outside surface of our body. However, it also means everything that's inside the body as well. So when you take a look at the inside of the body, you're talking about the lining of your organs, for instance, your digestive system, respiratory system, your genital system. So all of these basically are going to be covered or lined with epithelial tissue. Now, the thing is, your epithelial tissue will have a slightly different structure depending on where you are, right? For instance, your skin, which is epithelial tissue, is going to be a slightly different construction than the inside layer of your digestive system. Why? Because they're doing different things, right? Your skin is protecting you from the harshness of the outside world. There's a lot of mechanical stress on your skin, right? How many of you guys beat your skin up? It's easy to do. Just be alive, right? That's what you have to do, right? So as a result, the structure of that epithelial layer is different than, say, for instance, what's inside your blood vessels, because they don't have the same mechanical stresses that the outside of your skin would have. So the construction of it is different, but it's still the same thing. It's a liner for that particular part of the body. So that's a common sort of a thing. So your blood vessels, your cavities, by the way, guess what? The serous membranes, we just talked about that, didn't we? So the serous membranes, epithelial tissue, they're liners. They're lining the cavity. That's what they do, right? So think about it. It's physiological wallpaper. Not lovely, right? So generally speaking, whenever you take a look at this liner of epithelial tissue, the one thing you need to understand is it's got different regions to it. So for instance, if I were to take a look, and I'm just going to draw just a simple liner. So those are epithelial cells. They're connected together with adhesion joints or junctions. So they're fastened together, riveted together with these cell connections. Now, generally speaking, whenever you have a line of cells like this, you can't just like have them free floating in space. You have to have them sort of anchored or tethered to something structural. So there has to be some sort of minimal foundation on which you sort of attach them. And so the minimal foundation, generally speaking, is going to be right underneath, and this is going to be the basement membrane. Think of it like tack paper. It runs along the same lines as the connective tissue. It's kind of like just a sort of a foundation that allows you to be able to glue this line of cells onto it, so it's not just free floating in space. Now, what that basically means is that every cell, so if I were to blow this up, so there's my cell right there. So I would have this basement membrane, let me just say BM. And so typically your cells will have two surfaces. This surface here is what's referred to as the basal surface. That's basically toward the basement membrane. This surface here is what's called the apical surface. This is toward the free space. And then the lateral surfaces are on the edges. Now this becomes important in order to understand the nature of how these tissues are constructed and how to differentiate from one type of epithelial tissue from another. And so you have these two different pieces. You have the basal surface, which is connected to the basement membrane. You have the apical surface, which is not. It's the free surface on the other side. And then you have the cell connections, which are connecting the lateral sides or the lateral surfaces of the epithelial tissues. Generally speaking, when you take a look at epithelial tissue itself, it is what's called avascular. So that basically means there's no vessels in there. So things have to go into and out of the cell by diffusion. They just have to sort of diffuse in there or come out of it. And so oftentimes we will lose these because liners take the beating that the underlying tissues cannot take, right? So for instance, you do not want your muscles to take the kind of beating that your skin takes because, well, that would get a little on the messy side, right? It would also damage your muscles. So they have to be regenerated often. So you're constantly having to replace these cells. Now let's take a closer look at the basement membrane. The basement membrane itself has a couple of different layers. There's what's called the basal lamina and the reticular lamina. And so within the basal lamina, this is just a region of the basement membrane, you have two regions called the lamina lucida and the lamina densa. And we'll take a look at this here in just a second. But when you're taking a look at this, essentially what you see is you see a matrix of material that's made out of collagen, right? That's a protein fiber, toughness, strength, durability. Glycoproteins, which is your polysaccharide. Generally speaking, it's the laminin and also fibronectin. So these are the dominant types of glycoproteins that are there. And then proteoglycans, also a polysaccharide type of a structure as well. And so this is kind of part of what your basement membrane is. So really, it's kind of an extension of the extracellular matrix, so the ECM. Okay. And so this kind of helps give you a sense of sort of structure and foundation and integrity in the way that your epithelial cells, your epithelial layer is being built and developed and healed if you have to heal it, right? So it kind of creates a kind of a continuum for you to kind of work off, or a substrate to work off. Think of it as sort of like a canvas on which you paint or put your paint, right? So it's kind of like a continuous structure. So it's kind of what it looks like. So here's your epithelial cells. So here's your epithelium up here. And then when you take a look at your basement membrane, so here you have your basal surface on this side, your apical surface, which is open to the free space. And then when you take a look at your basement membrane, you're going to see that you have your different layers. So you're going to have your basal lamina, which is the first portion, broken into two layers, the lamina lucida and the lamina densa. Typically, those are histology descriptor names. If you're wondering where that comes from, that's a description typically from histology. So lucida means it's clear. Densa means it's dense, so more opaque in nature. So probably what happened was they got a really up close look at these layers, and they saw that there was one clear layer and there was one not so clear layer, and that's where these names come from. Okay, lamina lucida and lamina densa. And then those are resting on top of the reticular lamina, which is the bottom portion. And then oftentimes underneath that epithelial liner is going to be another foundation of connective tissue. Because typically speaking, your liner has to rest on something. It has to have some sort of a foundation, and oftentimes it's connective tissue of some sort. We'll take a look at the different types of connective tissues that you can put in there in order to be able to support this, this cellular layer. Okay, so let's go ahead and take a look at some of the things that epithelium does, right? So for instance, you can protect things. For instance, the lining of your mouth is epithelium. And so that's protecting the inside of your mouth. It's fairly thick because you are constantly subjecting it to mechanical stress. You know, even if you're just eating, it's constantly scraping and rubbing on the inside of your cheeks. Or if you're like me, you're a cheek biter, you yourself are actually putting mechanical stress on your cheeks as well, right? So it can be a barrier. For instance, skin is keeping the outside world away from your inside world because you are kind of a contained system designed to be at equilibrium within yourself, but you're not going to be at equilibrium if you're exposed to the outside world. So the skin allows you to be able to separate yourself from the outside world so that you can maintain an internal equilibrium and generate kind of homeostasis, right? You wouldn't have homeostasis if you didn't have skin to create that boundary. Sometimes it's you're going to use it for what I call physiological purposes. So you can use it to basically move things through them. So for instance, the nephrons in your kidneys are epithelium, a special type of epithelium that allows you to be able to move things through the cell itself. You can secrete stuff. For instance, your mucus glands are secreting mucus all the time. And you can also absorb stuff. For instance, your small intestines, which are absorbing nutrients, are also epithelium. So you can see your epithelium is actually not just a protective barrier. That's one function of your epithelium, but it can do a lot of other things. And I would argue those other things are probably more important than the actual barrier. So now when you look at classification of epithelial cells, one of the things we do is we basically classify them according to a couple of parameters. The first one is according to layering. So generally speaking, if our epithelial liner is only one layer of cells, then we refer to it as simple. So one layer on a basement membrane, and then that's it. Okay. Now, generally speaking, the way we define one layer is that one cell has to extend from both the basement membrane to the apical surface. So for instance, if we take a look at a cell, here's your basement membrane, the basal surface and the apical surface is basically identified in the same cell. Well, I am in contact with both the apical and the basal. That means I'm one layer, and that makes me simple. Now, what happens if you have more than one layer? If you have two or more layers, then basically you're what's referred to as stratified. And stratified, what that basically means is that one cell is in contact with the apical while another is in contact with the basal. So this is kind of what that looks like. So here is one cell here. I'm going to put another layer right there. So now you can see that cell number one is in contact with the apical surface. Cell number two is in contact with the basal surface. Two different cell layers that are in contact with the apical versus the basal surface. That's what defines these guys as stratified. In this case, they have two layers. Then there's what's called pseudostratified, columnar. And so this is basically one that appears to be stratified, but in reality, it's not. So the reason why it appears to be stratified is because, and we'll take a look at pseudostratified columnar. You guys probably saw that one on your trachea slides, right? So that was one where it looks like it's stratified. It looks oftentimes, when you have stratified, it looks like you have kind of two rows of cells, one row, a second row. In pseudostratified, it looks like you have two rows of cells, but in reality, there are cells in there that will actually have touch on both apical and basal. We'll see an example of that here in just a second. Transitional is a special type of cell. We only see it in the kidney. And basically what happens is it's a type of stratified epithelium, and so it changes shape. So for instance, originally it looks kind of cuboidal, maybe even columnar, like in the bladder. When the bladder is relaxed and it's empty, it'll have this sort of almost ovoid look to it, the cells. It'll look like this. And then when it stretches, it'll look flat. What we refer to as squamous, which brings us to our second classification. We can classify these by layering. That's the first one. The second one is we can classify these by shape. So when you classify it by shape, you can either be what's called squamous, which is flat and scale-like, and that's the one I kind of drew in my example. You can be cuboid, which, well, it looks like a cube, right? So this is kind of like the one I drew up in the top. So it looks kind of cubish-like. Or you can be columnar, which typically look kind of elongated and, well, columnar. And so you have these different shapes, flat cube or column, or you have simple or stratified in nature. And so this is kind of what it looks like. So here's your flat squamous cell, your cuboid cell, and your columnar cell. Now, when you take a look at these two, we're going to take a look at the squamous tissue. So this is an example of simple squamous tissue. So you can see it's flat. So these are flat cells that are basically one layer thick. So you can see the apical layer here, the basal layer here. It's the same cell. And so this basically makes it one cell layer thick. So this is simple squamous. This one here would be stratified. And this is basically because you can see multiple layers here. In that situation. So lots of different layers. So now let's go ahead and take a look at some of these. I'm going to blow these guys up. So here's our simple squamous. A lot of you guys were looking at the kidney as an example of simple squamous, and you were asking me, okay, where is it? Where is the simple squamous? So on the inside of the glomerular capsule. So I'm going to try to see if I can't blow this up here. Here's your kidney. So you can see right here on the liner, these little flat cells right here that kind of line this little white space, this little white cavity, those are simple squamous. They're flat, single layer cells, okay? And so they're kind of lining, and it's hard to do red on red. So let me try purple on red. So blow it up as best as I can. So you can see these little liner cells right there that are lining that little cavity. Those are the simple squamous. Yeah, you got to look quick, right? Otherwise it's going to disappear. So simple squamous are oftentimes hard to see because they're typically lining something else that you're looking at or paying attention to. And in addition to that, they oftentimes are very small as well. Now, simple cuboid is basically just that. So it's one line, one layer of cuboid cells. So for instance, in this particular case, you can see the kidney tubules are clearly simple cuboids. So here's the what's called the lumen, or the open region of the cell itself. So this is going to be where the basement membrane is, right here. That's the basal side. And then this side right here, on the lumen side, this is going to be the apical side. And so you can see that's true for the same cell. So that basically fits our definition of simple. So it's the same layer. This one is simple columnar. So you'll see this in intestines. So you can see, first of all, number one is these nuclei tend to line up into a nice, neat, orderly row. And so your basal surface is down here. Your apical surface is up here. Now, generally speaking, whenever you see something like this, that's basically a mucus pocket. And this is basically a goblet cell. So goblet cells are essentially these columnar cells that have this kind of glob of mucus in them. And what happens is they secrete that outside of itself, and they kind of create this sort of mucousy liner in the inside of your intestines. So your intestines are greased with mucus because a lot of the intestinal epithelium is secreting mucus into your intestines. That's to sort of make sure that the food that you've eaten and that you're currently digesting is sliding easily, no friction, through your intestines. Because the last thing you want moving through your intestines is friction. That would probably hurt a great deal. Okay. And so that's kind of what you have there. So that's an example of columnar. Now this is pseudostratified columnar. Now this one is going to be on the inside of your respiratory system. And so first of all, you can see that this is actually a terrible histology image, but you can see they're columnar in nature. You can also see these goblet cells, right? These little big globs of mucus. That are right here, these big blue globs of mucus. So those are goblet cells, and they're secreting mucus on the outside. The apical surface is going to be here, and the basal surface is going to be down here. And then this little blue line up here, this is cilia. So what this looks like is it looks like this. So here's your big glob of mucus, and then you have these little hair like cilia on the surface. That's why it's pseudostratified ciliated epithelium. Now, the reason why you have both the mucus and the cilia is because what happens in your respiratory tract is when you inhale dirt particles or smoke, what happens is this respiratory epithelium, which is secreting a lot of mucus, traps those particles. So it secretes this mucus. So you get this like little mucus layer, traps the particles in the mucus, and the cilia will constantly sort of bend back and forth, sweeping that mucus up into your cough zone in your respiratory tract, and you'll cough it out. Okay. So one of the reasons why smokers have that lovely little smoker's cough constantly, because their respiratory epithelial mucus is constantly trapping the smoke particles and trying to sweep all that out. Because your body doesn't think you're doing this intentionally. It thinks that you just happen to be in a smoky area and it's trying to keep your lungs clear. Okay. So the smoker's hack is actually the evidence of physiologically the smoker's body defending itself against the smoke itself. Okay. And so this is kind of what it looks like. So here's your pseudostratified ciliated epithelium. I want to take a look at this a little more up close. So here you can see that it looks at first glance to be stratified columnar, right? You kind of have like a row here of nuclei and a row here of nuclei, right? You have two rows, two layers, or you're thinking stratified columnar, but it's not. Why? Because when you take a look at it, what you notice is this guy right here is in contact with both the apical and the basal. That's simple. So is this guy right here. He's both apical and basal. So there are cells, and this guy in the corner, by the way, is also apical and basal. So the same cells in contact with both apical surface and basal surface, which is the definition for a simple layer, not a stratified layer. But it looks stratified because of its presentation. It looks like it has 2 rows of nuclei. That's why it's called pseudo stratified. It's actually not stratified. And that one, oftentimes, that's in your trachea. So that's the reason why I was interested in your guys is looking at the trachea slides, because that's where you'd see that pseudostratified ciliated epithelium. Okay. So that is mostly the simple stuff. The next group of epithelial tissues that we're going to get is going to be the stratified, the truly stratified, starting off with squamous first, and we'll look at cuboid, and we'll look at columnar, right? But we're going to start off with stratified now, multiple layers of these epithelial sheets. But since we're at our witching hour, this is where we're going to start next time.